Hello and welcome to the Learning About Learning lecture series. This is the 21st lecture, and we will be focusing on in this particular module we will be focusing on things that we haven't covered, that we said we will cover, and we have not talked about it, and that is pertaining to the course, and it's important to understand and get a broader picture of what learning is all about, right. As a first example, we will look at emotional memories. What are emotional memories and how these memories are compartmentalized in the brain, and how are they stored, and what are the nature of these organization of these memories. All along the course we have been talking about CS and US pairing, right. The where we take an animal present with a stimuli, a neutral stimuli, we call it as CS, and then pair it with a unconditional stimuli, unconditioned stimulus, as Pavlov called it, such as a shock. Now shock tells that, eventually this pairing happens such that CS by itself is capable of eliciting a response on its own without the US being present. Now in this scenario what we would like to ask is that what are all the different aspects that are present in this case, and where exactly and how exactly is the emotional valence of a memory stored. Okay. We know that the memory per se is stored, that you have a context. For example, we take a context that acts as a stimulus here, CS, and then pair it with a shock, and eventually when you try to place the animal in this context, then what you are actually looking at is the ability of the context to elicit a response in the animal all by itself. Okay. So now that this process we call it as learning, associative learning specifically, and we said okay, there is a response that has gone and with that obeys Rescorla-Wagner model, so on and so forth. But what we would like to ask is that where exactly is the emotional content, that is, see, the shock is a bad feeling. Where is that badness stored, or even it is not stored at all? Is it stored at all, or is something else that is being stored? More precisely, I would like to ask, how does, when an animal elicits a when a particular context elicits a response, such as context A here elicits a response of freezing, that is, now this is a fear inducing context, right. Now does the memory of the fear, right, that is the emotional here, emotional quotient here, right, that does it get stored separately than that of the context itself, right. One way to store is that you store the context memory separately, good. Second is, and then you store the fact that there is a shock of particular value proportional to the amount of current, amount of the degree of badness that the animal felt during that shock. You store them values proportional to that, and then associate these two things, these two facts, that this memory can elicit a shock, right. This is scenario number 1. Clearly here we are storing the information about the context separately and the value of the shock in a different manner, but and then the association per se, that's the key here. We the animal later develops an association, or all along it develops an association between these two memories. Other part is other way is very simple or a simplistic way to look at it is that you actually stored that the context memory. This is a bad context. All that you do is just it is a bad context, nothing else. Okay. Number 2, you just say that context A is a bad context, that is all. Now, these two are very, very different phenomena that can invoke different processes and can mean different things for behavior. Let us view this with an example, a practical example. Suppose let us say you go and land in a railway station of a completely unknown place or city in way past midnight, and it is pretty dark, and you are getting down from the railway station in the platform, and unfortunately you get mugged. Your wallet is stolen, your goods are stolen. That leaves a very bad experience in you, in your life. You tend to associate the railway station, the context associated with the railway station platform, the dark night and everything, and the new place to these bad feelings. So, one way to think of difference in these two scenarios would be, you remember this idea of the railway station platform, everything just that as it is. You also remember that you got mugged. It invokes a feeling, hey, these places I need to be careful, in future I can get mugged. So, that is scenario number 1. Or simply you just store the fact, a dark place with a platform around this where there is a moving train that comes in and it is night and you are getting down with hardly any people around, you are going to get mugged. It is just that you say that that is bad, railway stations in the night could be bad, that is all that you store. Now, why is that important here? Why do we need to know how are we storing this information? Let us say later on you want to modulate this response. You do not want to generically think all the railway stations in past midnight and with no people around could be bad. I mean, there are general pictures that you need to generalist pictures that you need to take in, but you do not want to develop a bad notion all throughout, right. You need to modify, you need to, so that you can actually exist. I mean, you can alter or have a response only for a really bad thing and not for necessarily for unnecessarily stimuli. Okay. So, in the scenario number 1, you would say that I just need to work on the association that has developed. In scenario number 2, it is a completely different thing. Every time the invocation of this entire context, you are invariably invoking the bad feeling. Now, suppose we know that it is not the scenario 2, it is scenario 1, then the mechanisms through which we can address this would be very different from the mechanisms with which we would address the scenario 2. It is a hard question to answer, partly because how do we even segregate and ask what the value from the memory itself. It is very intricate, right. You are associating a bad feeling with this context. So, how I am going to probe what is the value, is that value stored separately than the context itself. Whenever I am invoking both. I mean, that is the whole point of associative memory, right. If the scenario 1 is being there, whenever you are invoking that context, you would invoke that bad feeling. So, how do we even separate these two things? It as controversial, as important as these things are, there are always two sides to the story. People have been very hotly debating about these things for lot more time. Both the schools of thoughts existed. But now with the advent of modern technology and our ability to look inside the brain and say which of the neurons took part in which kind of memory, right, segregation of neuronal ensembles based on the memories that they are actually encoding, allows us to actually investigate this kind of stuff. So, how would you go about doing this? Michael Fanslow's lab at UCLA, they had used immediate early genes. These are the class of genes that we have seen little earlier in the course, which gets activated in response to a plasticity event in a neuron, right. So, they use this idea to probe if the memories of the context were to be stored separately than the association itself. Let us call this as the representation of the context, right. There is a representational memory, representation of the place or the things associated with the place other than the emotional quotient were to be stored separately than this association itself, the context and shock linked together, that linking. Then we must be able to identify two different ensembles, either in two different regions or in the same region or whatever the case may be, but we need to be able to identify two different ensembles. If we are able to identify, then we may be able to say, hey, look, they are stored differently, they are encoded differently in the brain. Now, to understand how they used the combination of immediate early gene and behavior to address such an important question, I need to tell you a little bit more about the associative memory itself and the experiments that we use to probe those associative memory, which are the contextual fair conditioning itself. We know that, let us look at the contextual fair conditioning itself. What is contextual fair conditioning? We are going to take an animal and then place them in a context. Okay. Now, the context here acts as a CS. In our example of the railway station incident, then it would be the appearance of the railway station, the place where it is, all together would represent the context. So, as soon as you place the animal in the context, or as soon as you get down of the railway on the platform into the railway station, you form a representation of this in your brain. Now, if we recollect, this is an event that is occurring in our life. So, it would be good to guess that this is a part of an episode, and naturally hippocampus, right, which we have been talking about quite a lot all these days, would participate in encoding such a memory, right. So, you would say that a contextual representation is formed in a hippocampus, that is hypothesis number 1, and where do we when that contextual representation gets associated with a US, in this case an electric shock for the animal, or in ours, for us it is the bad event of we getting mugged. Then that memory, the association of the contextual representation with the shock, the hypothesis number 2, years and years of research thanks to Michael Fanslow himself in his earlier days, has shown is stored in a region called amygdala. Okay. This is a region that is existing in the humans as a part of the temporal lobe, just at the very end of the hippocampus. In the animal, in the mice brain, if you remember, let us take one section of the the animal brain section. So, the hippocampus, if you remember our description of the Karl Laxley's experiment in the context of HM, we saw that this is the region where the hippocampus exists in the mice brain, and all along if you trace back you will see a bundle of fibers that are coming in and then making a bifurcation right down here, and this almond shaped region. Okay. That almond shaped region is called as amygdala. This is the region that has been known or shown to be responsible for encoding this association. Now, how do we know that? If we lesion out this region, the animal would not express fear, and there are equivalents of such brain lesions happening in humans, lacking emotional responses in these persons. So, that besides the point, we are now investigating here, what does the amygdala do? Does it store just this association, or it helps the other region to encode the bad the emotional valence along with the representation itself. So, what Michael Fanslow has shown is that in animals, when you place these animals in a context, depending on how quickly you give the shock, their ability to form this association will be affected. All right. So, now this is the time axis here, and we are going to delay the arrival of the shock with respect to the onset of the CS, and then for each of these delays we are going to measure how well the animal is able to make this association of context being bad. How do we do that? Once we train this animal, then the next day we come back and then put the animal back into this context and then see if it expresses fear. If it expresses fear, we interpret that as a context shock association has been formed. If it does not, then we say context shock association is not formed, and that is really the reason why the animal is not expressing any fear, and it is not freezing, thereby it is not expressing any fear. So, he did that, and he did that in multiple groups as a function, each group giving it for different delays. So, these were the initial days. People were still asking what exactly is the freezing behavior. So, he was actually measuring two things, freezing as well as defecation. Defecation is another behavior which we can as mammals tend to increase the defecation frequency when we are in a position of fear. So, what he is showing here is that as you keep increasing the delay in this axis, the degree of freezing increases, suggesting that mind it is the same amount of shock, same amount of duration, everything else is same. The only thing that is different here is the delay, clearly telling that unless or otherwise you give sufficient amount of delay for the context representation to be formed in the hippocampus. Remember, hippocampus is you can think of that as an integrator of various different modalities. So, all this information needs to integrate to form this representation of the context. Unless until you form that, you will not, the animal will not be able to form an association. What is that the animal can form the association with if the representation itself is not existing? So, in a sense, if you take the animal, put it inside a context, and immediately here is within few seconds you give a shock, next day you put back the animal in the same context, the animal will behave as if nothing has happened in that context at all, zero freezing. There is no memory of that context being bad in this animal. This is very critical. Remember this, we are going to utilize this in conjunction with immediate early genes to understand how Fanslow's lab has dissected out the different regions where these memories are stored. The bottom line is, if you there is a phenomena called immediate shock deficit, which is nothing but if you were to present a US, here the electric shock, right after the animal is introduced into the context without any delay, then the animal is not going to form an association with the context. Now, the first question you want to ask is, does the animal even forms a memory for the context? The answer is, you would like to say yes, it is forming a memory for the context, but then how would you go about testing it? Because the way we have been testing the behavior is through either freezing or some such form of a response, an emotional response, right. And if you want to dissociate that, then there comes the problem, and this is exactly where the immediate early genes came in handy, where now you do not want to look, you do not have to look at the behavior of the animal to say whether the memory is formed or not, but actually you can look at the neuronal ensemble and how they are getting activated to answer the question of whether the memory is being formed or not. Just to recap on our immediate early gene, these are the genes that gets expressed as a first response as a to the various different signals that the neurons are getting. In this case, we would like to look at it as a stimuli that the neurons are getting. So, there are different classes of immediate early genes, and even with effectors and effectors, if you remember, and even within the same classes, depending on the size of the gene and the function that they are associated with, their expression dynamics or expression kinetics, the time scale at which these different things can happen, vary. And they have used that kinetics here to dissect out this question. How would you use that? Now, let us say if we know, I mean for in this particular in this particular case, the immediate early gene that they are using is called as ARC, and ARC is known to express or ARC mRNA is known to be expressed in the nucleus in the initial 5 minutes, which then gets exported out of the nucleus to the cytosol after about 20 minutes. So, what they are going to look at what is the following. They are going to look at two different events, event 1 and event 2, and you are going to look at then say which are the neurons that took part in event 1 and which are the neurons that took part in event 2. So, how would you differentiate? Let us look first at the figure B here. Okay. When you look at the, so this circle, if it were to be the nucleus of the neuron, so if you look at this circle, okay, colored green here to identify the nucleus, you would see two puncta, and these puncti represents the transcriptional activation sites in the neuron, and you will see this within 5 minutes of the activation of the neuron for ARC. While if you were to look 30 minutes later, these guys diffuse out. As a result, what you see, and these and many more of these guys diffuse out, and as a result you see, what you see is that you have higher probability of locating these red structures outside of the nucleus. So, by characterizing a given neuron to say that, hey, these red structures are present outside of the nucleus or inside of the nucleus, you can say if a given neuron is encoding, a given neuron is actually encoding event 1 or event 2, because if this neuron were to encode something, it had to encode something that had happened 30 minutes earlier. So, it has to be a event that has happened before. and this neuron represents those events that had happened just then, like 5 minutes before. Now, let us look at the behavior. So, what they did were two kinds of things for event 1, a delayed shock group, which is nothing but the regular contextual fear conditioning. You expose, take the animal, put them into the context, wait for a few minutes, then present the electric shock. Group number 1. Group number 2, everything remains the same, except now you give the shock immediately within few seconds. We know when you test these two groups, the first group shows freezing, shows an association of the context with the shock, while the second group has not had the ability to make that association, because the context memory itself was not formed during the time when the shock was given. So, there is nothing for it to associate with in terms of the context. So, in a 20 minutes test, a test that is done 20 minutes after this event 1, two kinds of event 1s, you can actually measure the behavior, and when you measure the behavior, that is the extent of freezing you see in the delayed fear conditioning case, and as we expected, the immediate shock group does not show any freezing. Clearly, there is no emotional memory that has been formed or that has been associated here. Now, let us go ahead and look into the hippocampus of these two different groups. Now, when we do that, what you will see is that three different kinds of cells. The green here identify the nuclei. This is called this is a specific kind of a stain that identifies the nuclei, and they are actually looking at the hippocampal region here. And let us take an example of this cell. Clearly, you can see the green and the red color. Red color represents the ARC mRNA transcription. Both of them are present. That is, the red is present where the green is. So, you would say they are nuclear active. There are also other neurons where there is just green, there is nothing else, no red at all. I am just pointing out to the bigger circle here, no red at all inside them or surrounding them. So, clearly those nuclei do not express mRNA, the ARC mRNA. And there are other cells such as this, or there are other spots of red, I should say, where you do not see any green at all, like for example here. So, they are outside of the nuclei, right. You call them as cytosolic mRNA, and good example of both being present is something like this, where you see red sphincter on the green and there are some red outside of the green, but still inside in the vicinity of the nuclei. So, you would associate with that cell. Now, this allows you to classify the neurons into three different types. Type 1 are the neurons that are present, whose nucleus is very clear. So, you call them as cytosolic expression. The neurons whose nuclei contain the red puncta and just that alone, nothing else, nothing around the cytosol, nothing around the nuclei. So, you call them as nuclear only events, nuclear only neurons, and the neurons that have both the cytosolic as well as the nuclear. You call them as double label neurons, and you can clearly say these are encoding different things. These guys, because the nucleus is clear, nothing has happened just before the brain was extracted. So, things must have happened, the arc mRNA that you see here must be because of some things that has happened before the extraction process, quite before the extraction process, 20 minutes before the extraction process. So, they are most likely the event 1 neurons, while the nuclear ones just then are expressing the mRNA. So, they are quite likely are transcriptionally active in response to event 2, while the double just by the same arguments would be representing both 1 and 2. Good. So, now let us look at the hippocampus of the group which underwent immediate shock. What we see here is that very little of the neurons are purely event 1 neurons, very little of them are event 2, but a large fraction of them express both event 1 and event 2. That is, remember, event 1 is the training event and event 2 is the retrieval event. Okay. So, all those guys who participated in encoding the contextual memory at the training do get activated at the retrieval. However, when they are during the testing, but then when you remember this group, the immediate shock group did not express any fear when you are actually putting them into retrieval. The freezing was close to 0. So, there is no fear expression here. This is not due to fear expression. Still you are able to see considerable fraction that are active in the hippocampus for both training as well as for retrieval. Strikingly, a similar amount and not any more you see in delay conditioning, even though in the delay conditioning you do see an expression of fear. So, just by looking at the neuronal population at hippocampus, what you would say is that you will not be able to tell which of the group got a shock or which of the group is expressing the fear and which of the group they are not expressing the fear. Both the groups got the shock, only the reason why the other one of them is not expressing is because they got the shock very early, the immediate shock. They do not associate, they do not express the fear, they do not think that the context, the training context is fearful. On the other hand, if you do look at the hippocampus, if you do look at the amygdala, what you see is this beautiful dissociation. In the immediate shock, you do not see the double events at all. There is a set of neurons that are getting activated for the first event. There is another set that gets activated during the retrieval event. The association of the it is completely different, and the double label which would represent the memory here, right, truly, that is those guys that were active during the training were how many of them are active during the retrieval. If you ask them, is negligible, 0. On the other hand, in the delay conditioning, what you see is that almost nothing at all during the training here. On the other hand, if you are asking for the retrieval based, after the retrieval what happened, you see a considerable amount of fraction of neurons that are coming on, while all of the neurons that were active during the training were also active during the retrieval. That is why you have the double labeled. Let me repeat again here. The neurons that encoded only event 1 is almost nil. Every single neuron that were active during the training were also active during the retrieval. In addition to that, there is a small fraction that were active selectively during retrieval. Keep that in mind, we will come back to that. This is in direct contrast compared to the hippocampus, suggesting that amygdala really is encoding the valence or the value of the emotion here, and while the context per se is encoded in hippocampus without any value of the emotion associated with it. Then what do these neurons represent? The neurons that came online, that got activated during retrieval. They were not active at all during the primary event, the training event. What are they doing here? With that thought, let's keep that thought in mind. Let's look at another phenomena here, the phenomena of second order conditioning. Okay. Before I go there, I just want to repeat the conclusion here of the slide. Hippocampus, from these experiments we know hippocampus stores purely the representation of the context, while amygdala stores the emotional value associated with that context. And it is the invocation of both that results in a behavior or expression of the fear associated with the context. In some sense, you could intervene such that the fear associated with the context at that's stored at amygdala could be suppressed either through infusing a drug or selectively making the drug go to the amygdala. Thereby you can retrieve this contextual memory without the fear associated with it. In this case, they used the behavior itself, the training protocol itself, to dissect out these two along with the immediate early genes, but in essence that is the point. We tend to store the memories of the context or the episodes of the event separately than the emotional valence associated with them. Now let us look at the second phenomena, second order conditioning. We have talked about the second order conditioning quite a bit during the reinforcement learning. Okay. Now what is second order conditioning? Let us understand first the primary conditioning itself. We know that. So we take a CS and then pair it with the US over a period of time or over a repeated presentation or even on a contingent presentation. You end up developing a behavior in an animal, a new behavior in an animal, such that presenting the CS alone elicits a new response, a CR, right. So this behavior was not there in the animal before the training. That is the whole point of this learning, right. You did this exercise to generate this behavior. Stimuli alone can elicit a response. Now the beauty of this is that things do not stop there. Now that you have this stimuli, a conditional stimuli capable of eliciting its response on its own, right, after the training, right. Can you use this to pair it with another conditional stimuli that does not have a response on its own till now, such that the new conditional stimuli develops a response on its own? If you do that, that is what second order conditioning is. Let's see what it is. What is it again? We have done this conditioning. We have acquired a ability in an animal to. We have made the animal acquire an ability such that a new stimuli until now is passive is able to elicit a response in the animal. Now I am going to take that stimuli, that CS, that trained CS, and then pair it with a new stimuli, CS2 here. Okay. This does not have a response on its own. This has, thanks to this training, because of this training, this has a response. This does not have a response on its own. Now if I pair it, what we see is that you can actually later present just the CS2 alone eliciting a response. Now again This response need not be same as the CR of the first one, but in general we tend to choose a similar stimuli. So, the responses tend to be same. So, the idea that you can take a paired stimuli that acquired a new response and then in turn use that in training another stimuli, this whole phenomena we call that as second order conditioning, or in short SOC. This is beautifully illustrated in a very simple laboratory setting. All right. So, I will talk about that example and present you with some of the videos, and then talk a little bit about social learning and what we have done in our lab in relation to the social learning in the final lecture of lecture 22. Thank you.